You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast. I have Assistant Professor Donny Hanjaya-Putra. He's in the Department of Aerospace and Mechanical Engineering at Notre Dame. Also, Assistant Professor concurrently in the Chemical and Biomolecular Engineering Department, College of Engineering. We're going to be talking about stem cell therapies. So, Donny, thank you for coming.
Well, thanks for having me in this program.
Yeah, so tell me about the work that you're doing on stem cells. What does it involve?
So we in the lab is interested to utilize engineering principle to control stem cell differentiation and try to use it for, particularly to use it for tissue regeneration and tissue engineering. So our lab is at the interface between engineering and medicine. So we like to use this as a way so we can control the differentiation in a way we can model and treat different diseases.
Okay, so any specific diseases that you're focused in on? What kind of modeling are you doing?
Yeah, so we are particularly interested in diseases that associate the cardiovascular lymphatic system. So anything that has a blood vessel and lymphatic vessel, those are particular throughout your body. So the cardiovascular system is the one that is regulate your blood, and the lymphatic system is the one that drains all your fluid back to the heart. So those are important to maintain tissue hemostasis as well as different nutrients inside your body. So any defect, any disorder in that system will cause different diseases. It could lead to peripheral diseases like limb ischemia, or lymphatic disorder could lead into lymphedema, so swelling of the limb.
How does this, so lymphedema, ischemia, is that what you're trying to model the action of stem cells in those conditions, or I don't understand?
Yeah, so I'll give you an example here. So stem cell is it's a cell inside our body, and particularly in the circulating blood, we know that this cell can give rise to the blood stem cell as well as the vascular stem cell. As we as an adult, we have this stem cell population that can give rise to regenerate. And if you have a vessel defect, this stem cell can repair through it. But if you have a chronic disease such as cardiovascular disease or diabetic or aging, this number of stem cells is becoming lower. As we age, the number of these stem cells becoming lower, and as well as the ability for them to regenerate also lower. So they have a defective in the stem cell. So what we're trying to do in the lab is, can we use engineering principle to kind of rejuvenate or kind of regenerate the cells so they can become like normal, like a healthy cell, so they can in fact become degenerative tissue again.
So what everyone experiences over time, the number of stem cells they have in a given tissue decreases. But what about the efficacy of the remaining stem cells? Does that also go down? And how can we measure that if so?
Yeah, those are also goes down. So, give you an example, like if you expose this stem cell to high sugar content using the patient with type 2 diabetes or type 1 diabetes, because there's a lot of high blood sugar in the blood, so it causes defective in this stem cell. So there's epigenetic regulation that causes this cell to be defective, and the number is down as well as the efficacy of this cell is down.
Using epigenetic changes,
Yeah,
you believe, are what leads to the reduction of stem cell populations or the reduction of efficacy of stem cells?
right. So we have some preliminary data that show that there's some epigenetic changes that cause this defect, which could also be what we're looking for in the lab now is that can we use a small agent such as either a small molecule or therapeutic drugs that can be used to kind of interfere with these epigenetic changes so that we can correct this cell back to the normal condition.
So, okay, if epigenetics are the mechanism that you think that changes the stem cells, why not look for a solution that, or the reasons for the epigenetic changes and maybe how to reverse them instead of a small molecule drug? Why look for a drug to interfere or change the function of something?
I mean, well, a small molecule is nice because you could take these drugs as you could give this to patient and they can take this orally, right? And if this small molecule can have a downstream signaling that can inhibit certain pathway, then we could correct those epigenetic defects as well. So,
but if we don't understand the full method by which the epigenetic changes happen and what the changes are, it might be hard to create a drug that would reverse that. I mean, for instance, has anyone looked at, let's say, a particular type of stem cell that has a very short lifespan and track the epigenetic changes that happen, the expression changes that happen within that stem cell? So if we take two types of tissue and one has very short-lived stem cells and one has very long-lived ones, and we compared those two situations, for instance, to see the difference in efficacy of the stem cells over time in those two kinds of tissues, has that been done?
Yeah, so that's been done, the comparison either how this healthy and disease cell population differences in terms of the epigenetic, either that one as a metabolism, how they metabolize different nutrients. So those are done, and we know in the lab is we could use a small molecule that interfere with some sort of cascade to kind of correct the cell back to normal phenotypes.
Okay. So at what point would you want to work with someone when they have a disease or just as young as possible? I know there's some companies that are trying to harvest stem cells from a person when they're younger. Like, what's your method by which you want to help people?
So that's a great question, right? Because now cord blood banking is becoming a pretty common procedure in the developing world, right? So what if you could bank this cord blood in a way that we could store this cord blood stem cell that eventually you could use this for if your baby become adult and at some point during the development there's some diseases, can you use the stem cell for those diseases as well as for other relatives, right? So I think the earlier the better, so that you could use it early interfering to restore those. Also, we work with this cord blood now that we know that you could not only use the stem cell as to regenerate, but you could also use it to predict future health outcome, right? So, for instance, if we store this cord blood stem cell and you have a healthy baby that was born from a mother who is an uncomplicated pregnancy, compared to a mother who has a complicated pregnancy, either those are through gestational diabetic or through preeclampsia. So those cord blood are different, right? So the baby, the infant was born from a healthy mom, typically have a normal stem cell, but the baby, the infant that was born from mom who has preeclampsia or gestational diabetic, those stem cells are different. We know a lot about how this complicated pregnancy affect the mother, but we do not know a lot about how this complicated pregnancy affect the infant, the offspring, right? So if you could use this kind of stem cell to predict how this baby will grow up, whether this because of exposure to early hit early on during their when the baby, it causes this infant to have develop higher risk for cardiovascular disease or metabolic syndrome later on. Those are unknown, right? So can you use the stem cell to predict that as well? I think those are pretty exciting new field.
So what kind of models are you studying stem cells in? Are you able to study them somehow in culture, human stem cells, or do you have to use rat stem cells? Like, what's your lab model?
So we like to use a human patient derived cell. So we collaborate with pediatricians, pediatric down here in Indiana School of Medicine. So they isolate this cord blood stem cell from a newborn baby, and we take this tissue sample and we culture them in the lab and compare if what from a normal pregnancy and compare this to complicated pregnancy that could be from either diabetic, gestational diabetic, or either preeclampsia, and others differences in the function of this two stem cell. Now, this stem cell also secrete this small exosome. Exosome is a small vesicle, so this cell secreting this vesicle is a small particle inside the cell. Now, inside this particle, it contains a lot of different information. So it could be either small RNA or protein, right? So those are something that interesting, becoming more interesting in the field because now you can compare this expression of different exosomes and see if you could predict a difference as a biomarker to see if there are difference in these two populations.
So all right, so stem cells in particular, well, not just them, but all cells, I'm sure, but stem cells,
Correct.
You've looked and you've seen they produce exosomes, little fluid-filled vesicles. Have you been able to get an exosome and characterize exactly what's inside of it? Do they tend to customize and custom package exosomes at different times that have different things in them?
Yeah, so there's I think this is a hot topic recently because it seems like this is a small particle that was secreted by the cell. The hard part about that is there's a lot of different protocol in a way to isolate this exosome and analyze them because you could imagine that this is a small particle, so you have either to centrifuge it at high speed to kind of concentrate this nanoparticle, and then or use a different chemical gradient to kind of isolate them typically. So once you have those, you could do a different analysis. So you could do typically a proteomic to figure out what kind of protein are there. If you're interested in RNA, microRNA, we typically do RNA sequencing to figure out what is the microRNA inside there. I mean, microRNA is a pretty interesting to look into because not only that this cell are secreting exosome that contain microRNA, and they use this for cell-cell communication. So they in a way that it is secreting this microRNA, and this microRNA inside this exosome can go to the next cell to kind of regulate the next cell as a way for them to communicate. So if we know that if a disease cell or stem cell that are exposed to this chronic disease secreting this microRNA, perhaps this microRNA can tell us something about why, and we can use this as a biomarker in the future. So,
what is the size of an exosome on average, and what is the size of a human cell on average?
Yeah, so the exosome in the field we call it extravascular vesicles. So there's a big range of this size, right? So ranging from 100 to 200 nanometer. But what we call exosome is something that's smaller than that. So it's around typically around 10 to 50 nanometer of this small exosome. And the cell is typically in the micron size. So this is a small particle that is by the cell.
Right. Okay. So, and I guess, cells put out exosomes, but they also take them in. And maybe this is the method of cell-to-cell communication, right? Or one of the methods.
Correct. So there's a lot of literature out there on cancer. Cancer cells secrete this exosome in a way for them to communicate to other cells and to a way to spread their message to other cells. And now we're starting to learn that stem cells do the same thing, that they secrete also exosome. And inside this exosome, there's a microRNA that regulates other cells. So in a way, can we detect this microRNA and do a rapid screening to see if we can use it as a biomarker?
And I guess profiling how the exosome expression or the exosomes change over time would give you a lot of clues maybe as to what's going on.
Exactly.
It's so complicated and interesting, but it's amazing, you know.
Yeah, I think it is a hot topic, and a lot of people are working on this field. So it's an exciting time.
Well, so what are some of the new discoveries or surprising things that you've learned about stem cells in your lab?
Yeah, so I guess going back to the first topic that we talked about earlier, that what if you have this cord blood stem cell derived from either normal patient or patient, a mother who has chronic disease. In our case, is diabetic. So what we found is that if you were born from mom who is a diabetic, your cord blood stem cell has lower ability to proliferate, and they have a defective their ability to form this vessel network. So we found that there's a particular protein that is highly expressed in the cell, and typically this expression of this protein is not as high in the normal stem cell. So because of this high expression particular protein, they are not able to proliferate as well, and they're not able to migrate as well. Consequently, because of those, they cannot form blood vessel as well, right? So the strategy that we have in the lab is, can we use small molecule to kind of correct this? So because we know the pathway, the signaling pathway, we could use a small molecule inhibitor to kind of inhibit this protein's expression of this protein. By downregulating this protein, we could effectively reduce this protein level, and consequently, we could kind of recover their phenotypes, so they become like a normal cell, right?
We mean now, they become a normal cell?
Yeah, so we can rescue their stemness. They can rescue their phenotypes.
I mean, anytime they're going to repair, or anytime they do their job, essentially they give up their old job of being a stem cell, and now they've finished their differentiation to the specific tissue that needs repair, right?
Correct. So we're talking about progenitor stem cell. This cell are able to give rise to a daughter cell, right? And typically, this daughter cell have less proliferation ability. But what we know, though, even the stem cell from the disease stem cell, the ability for them to proliferate is less compared to normal one. So if you could rejuvenate the cell, then they can still proliferate as well as the normal stem cell. Does it make sense?
Okay.
Yeah. So you're referring to the dead daughter cell. Once they proliferate, the daughter cell has less ability to proliferate, but it's still their stemness is compared to normal, there's less, right? So, and if we can find a way for us to kind of rejuvenate the cell, then we could still use this disease stem cell, and it can rejuvenate those progenitor properties so they can regenerate the tissue.
Well, what about looking at the different populations of stem cells? I've heard in some organs and tissues, the stem cells very rarely divide, and in some they divide constantly, like hair, nails, that kind of stuff.
So you're talking more about tissue-specific stem cells. So those when we are born, the stem cell lose the ability to become pluripotent, to regenerate to other cell type. But as an adult, we maintain this small amount of tissue-specific stem cell that can regenerate, right? And you mentioned about the gut and the nail and the hair and the skin. They still maintain some tissue-specific resident cell stem cells that they are quiescent. In a way, they are quiescent, but when there's a signal from the outside, the signal will secrete uh, different cytokines so that the cell will able to now mobilize and proliferate and generate new daughter cell, and those daughter cell can repopulate to regenerate the tissue, right? So those cells are very rare. You mentioned about the one in the skin, the one in the gut. So those are very rare. We also know the one there's in bone marrow. Those are a blood stem cell, and what we current working is also the one in the circulating blood, and we found the cord blood as well in the peripheral blood. There's a small population of cell that can also give rise to endothelial cell. So those are the cells that we're working with in the lab. So
are you working with cord blood cells, or are you working with adult stem cells?
We're working with cord blood cell.
Okay, so these are still pluripotent cells. They haven't differentiated at all.
No, the cord blood is already an adult, right? Because once they're born, so those are adult cells. So this is not pluripotent. This is a regenerative cell. So this cell can only give rise to the blood cell and the endothelial cell.
Oh, okay, I see.
Mm-hmm. Yeah.
So they are different. So you're studying the stem cells in blood and the stem cells in the endothelium.
Correct. Yeah.
Okay. So what have you found that's unique or interesting about them so far in your studies?
Yeah, what we found exciting is that you could rejuvenate the cell using a different molecule, right? But now this molecule, if you want to use it for cell therapy, then in a way you could have to inject this cell, and then at the same time you have to give this small molecule inside the circulation, so in a way give the drug. So so you have to this to concurrent therapy, the cell and then the molecule. So what we thought recently is that rather than doing this co-therapy, can we put this small molecule inside on the surface of the cell? So I think the analogy that I like to give is that in this field, people start talking about putting nanoparticle in the surface of the cell. So putting like a backpack into the cell. So we send the analogy that we like to give is that we send soldier to the battlefield, and they're carrying backpack, right? Inside this backpack there's a walkie-talkie or their medicine, right? When the soldier is smart, they know where to go, but we give them some equipment so that they can survive in this battlefield. Similarly, this stem cell, they know where to go inside your body when you inject them. So, but from this disease phenotype, we need to rejuvenate them. We need to give them some sort of bioactive molecule. So what we put is we put a backpack into this cell. Inside this backpack, there's a small molecule and there's a drugs, so that when the cell needs it, this nanoparticle can release this molecule that can be used by the cell at the site of injury.
Well, they do that. That's what they do naturally, you're saying, or that's what you want to do with the addition of other types of drugs?
No, they don't. I mean, we do this ex vivo, so we take the cell out and then we modify in the lab and we inject this back in vivo.
So you're saying you're using the ability of stem cells to very precisely target certain areas of a damaged tissue, and then for them to be the carriers of small molecule drugs that get released. But this would prevent a systemic release. It would make a very targeted release of a given drug.
Correct.
That's pretty smart. I like that.
Yeah. So we kind of use their I mean they when we inject the cell, they already know where to go. So in a way, it can be used. We hijack the cell, or we put a backpack inside the cell so that we could give a small molecule drug to where they go. So
that's really interesting. Have you tried this yet? Have you taken drugs that are currently in use that are known to have an effect, and given them to your carrier pigeon stem cells to take to the tissue?
Mm-hmm. Yeah. So I mean, we're not the first one who did this. I think this idea has been there in the field. I think people have been working with hematopoietic stem cell, and I think there's a group in MIT who did this, and is a pioneer on working on this. And his group has been showing that you could take hematopoietic stem cell, and there's a stem cell transplantation, And in addition to this transplantation, typically patients are taking any adjuvant drugs, right? So drug that has to be taken in concurrently with this stem cell transplantation. So the idea is can you deliver this drug together with this stem cell? So those are done in hematopoietic stem cell, and we currently are exploring the same idea and looking at cord blood stem cell. So can we use this stem cell that can give rise to a new vessel in a way that if you inject the cell, they can go to the site of injury and repair the damaged endothelium. So can we also put some sort of drugs so that it can also go to the site of injury with those drug carrier? So we've done this in vitro assay, and we show that it can work in vitro. We now currently looking into different in vivo models, so we can show that it's a bit effective in vivo.
When you package a small molecule drug with a stem cell, it goes to the right place. Mm-hmm. But how do you time it so that it releases its payload at the right time? How do you signal that?
Yeah, so that's a great question, right? So that's part of the engineering design, right? So now you start talking about how do you design this nanoparticle so that in a way you could release this small molecule at the right time. So there's multiple ways you could do it. We could use this nanoparticle we like to use a lipid nanoparticle, so it has a phospholipid, so it has a hydrophilic and hydrophobic tail, and under certain conditions they start forming this micelle that form self-assemble into nanoparticle, right? So if you could engineer or somehow use a different design, or how big is this tail of this nanoparticle, if you could add regulation of this nanoparticle, if you add different chemical head group, that can affect the thermodynamic of this nanoparticle, and in a way also affect how the drug being released from the nanoparticle.
Well, this made me wonder. Whenever we take a drug, I don't even. It's not really talked about, but I would think the body sees it as a foreign substance, so it would mount an immune response, it would mount an inflammatory response. Is that the case? And if so, what do you do to stop the stem cell from being attacked itself?
Yeah, well, I think when you talk with taking drugs, there's multiple different drugs, right? So I think the simplest one is small molecule drugs. Could be aspirin or either different agent. So those compound is small enough that can be effective at the site of the binding site of different on the cell, but it doesn't really affect your immune response. So the small molecule typically have very low immune response. So this is why probably pharmaceutical company like to work with small molecule. Not only it's easy to synthesize, but also pretty low immune response. Now, when we start talking about biologics, so those are involved any protein, drugs, as well as antibody. So those are larger molecule, right? And typically, this protein and antibody drug are isolated from either cell or either a mammalian cell or mammalian cell or bacterial cell. Because of that, then they have some immunogenic response. So if you inject antibody drugs or protein drug, typically have some sort of immune response, right? So, and now we're talking about cell therapy, which is on the other spectrum, which is much bigger than any of this molecule. So those cells typically give a bigger immune response, right? So this is why we like to use autologous cells. So if you use your own cell, then it doesn't have an immune response, right? So if you use a cord blood, so cord blood typically is your own cord blood. So it's autologous stem cell therapy. So hopefully those give you the lowest immune response.
Okay. I didn't realize that small molecules much less immune response than large ones. Interesting.
Yeah, typically the small molecule is, this is when we're taking aspirin, taking vitamin D, so those are pretty small. And you take this, typically you go to your liver and your liver metabolize this. So, and if you're taking antibody or protein drugs, so typically these drugs are using IV injection, right? So you cannot take it to your gut because the acid in your gut will neutralize this antibody or protein right away. So antibody or protein drug typically are given using IV or subcutaneous, right? So once they hit inside the vessel, then you start seeing all these white blood cells looking at this and, well, this is a foreign molecule. We're going to attack them. So
I just think that's fascinating that the stem cells, again, can take these small molecule drugs to very specific places because, again, this is just my theory, but there's a lot of redundancies in the body and a lot of redundant receptors and things like that. And it seems like the problem of a lot of small molecule drugs is that they act systemically. And if they acted locally in the area we wanted them to act, they would have much less side effects. So this seems to accomplish that.
Yeah, so I think that's a problem for any of these drugs, right? So either those are small molecule protein or biologics. So they have a very narrow therapeutic window. By therapeutic window, I mean they can be effective at a very small dosage, right? So, but you have to take this at a pretty large doses orally, and it go to the circulation, and the systemic level is pretty high at the end, right? So, and that can cause side effects. So, any way we can reduce those systemic level becoming low, and in that could pretty beneficial for the drugs because now you could start working in a larger therapeutic window, making the drug more effective but doesn't cause side effects, right? So.
Another thing I thought of is that if you're able to target a specific area, you could probably use far less of a given small molecule drug because you don't have to account for diffusion around the body. So you can use much lower doses and get a better effect.
Yeah, that's exactly right. So if you could kind of localize this drug at the site of injury, then you don't need a lot of dosing, systemic dosing, because you could localize it at the site of injury, and you could concentrate most of this drug to be effective where it needed, so it doesn't cause any side effect anywhere else in the body.
Yeah, how much less? You might be able to use what, like 1,000 times less or 10,000 times less? I mean, it seems amazing that how much more effective this could be.
Yeah. I mean, I think the most, I guess, the big example is anticoagulant drug. So this drug is heparin or aspirin. So all this drug is very good in preventing clot, right? So they work very well in preventing blood clot. But at the same time, the highest circulating concentration of this drug, heparin drugs, can cause side effects, right? So you can cause bleeding, right? So you could see a lot of lawsuits because of how this drug works so good, but at the same time can cause bleeding, which is pretty dangerous. So if we could find a way to kind of localize this therapeutic this drug so that it can only target where it needed, then in a way you don't cause all these side effects.
Yeah, this is like the Uber, using stem cells as Uber to get drugs.
Yeah, that could be. Yeah.
Well, that's really interesting. Is there a name for this field or specialty, or is it just modifying stem cells for a purpose?
Yes, I mean everybody has their own favorite name. You will see they call it cell surface engineering or therapeutic cell surface engineering. There's stem cell engineering. So in a way, they like using engineering to modify the cell, using different bioactives to put this conjugate into the cell surface.
Okay, interesting. Well, Donny, this has been a great call. What's the best way for people to find out more and to get in contact with your lab or you and ask questions?
Yeah, I think it will be. This has been great discussion as well for me. You can reach out to me. I have a website online, so it's dhplab@nd.edu. So you could reach out to check the website. Typically, I try to keep it updated at that website, as well as my email address is dputra1, dputra1 @nd.edu. You could also follow me at Twitter. It's a DHP group.
Well, very good. Well, Donny, thank you for coming on the podcast. I appreciate it.
All right. Thank you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier, Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.